likelihood framework for B-cell repertoire phylogenetics. Please.
Thank you. Can everyone hear me? Okay. So, hi, I'm a postdoc with Steve Kleinstein's group at Yale, and I'm here to talk about my work with B-cell repertoire phylogenetics. For B-cell repertoire analysis, the molecule of interest is antibodies. These are proteins released into the blood and mucosa that neutralize pathogens by binding to structures on their surfaces called antigens. This marks the pathogens for destruction by other cells or effectively binds to their surfaces and neutralizes them. This mechanism depends on the fact that antibodies have structures which match structures on the surface of pathogens, which leads us to the question of how exactly do antibodies recognize the vast diversity of pathogen structures that humans are exposed to? And the answer to that is essentially an evolutionary one, and it relies on the fact that antibodies begin their lives as receptors on the surfaces of B cells, creatively called B cell receptors. B cell receptor genetic diversity is produced through a two-step process, the first step of which is called VDJ recombination, which is essentially when a new B cell is produced, the gene which encodes the B cell receptor shuffles itself and is spliced back together, which produces a more or less unique rearrangement per B cell. And this produces a unique B cell receptor for each new B cell which is produced, creating a large and genetically diverse population of B cells capable of anticipating a wide array of pathogen structures. These, however, are really just random guesses, and in order to be effective, B cell receptors need to be refined, and this refinement is essentially a process of evolution by natural selection. When a B cell binds to its cognate antigen, it is activated. It divides very rapidly, it mutates very rapidly in a process called somatic hypermutation, and its descendants survive based on their binding affinity to their cognate antigen. This causes an increase in binding affinity over time, but it also creates a clonal lineage, of B cells which can trace their ancestry back to a single naive ancestor. And this means that phylogenetics is a pretty obvious tool for understanding B cell receptor genetic diversity. And it's shown to be quite useful in a number of applications already, particularly in kind of obviously tracing the history of a clonal lineage, but also in predicting unobserved intermediate sequences of clonal lineages, which is very important for antibody engineering, but also just understanding the types of mutations that shape the diversity of the B cell repertoire in individual people. However, there are some things about B cell biology and B cell repertoire data that should give us some pause in applying standard phylogenetic techniques. Chief among them is the fact that model-based B cell or model-based phylogenetic trees generally are built off of Markov substitution models, which are built around the assumptions of independence of nucleotide site mutations and the fact that, or the assumption that substitution rates are symmetric. So, for instance, A mutates to T at the same rate that T mutates to A. So almost all phylogenetic substitution models are built around these assumptions. However, what we know about somatic hypermutation in B cells doesn't really gel with this. In contrast to a lot of systems, mutation in B cells is actually driven by a set of enzymes, chief among them AID, which operate on particular hotspot motifs. For instance, WRC, which could be an example of AGC, mutates much more rapidly than most other motifs, whereas SYC mutates much less rapidly. And this means that somatic hypermutation is actually context dependent, which violates the assumption of independence among sites, and it's non-reversible because certain motifs decrease in frequency over time and certain motifs increase in frequency over time. And because of this, we previously introduced the HLP17 substitution model implemented in IgFIML, which weights codon substitutions by the probability that they occur within particular motifs that have been pre-specified. And this has its own set of assumptions, so it's not a perfect model, but it's a step forward. Another issue is the fact that B-cell repertoire data is quite different from the kind of data that we typically deal with in phylogenetics. Generally within phylogenetics, we have one multiple sequence alignment from which we estimate a set of substitution parameters and a phylogenetic tree. With B-cell repertoire data sets, we usually have hundreds to thousands of different multiple sequence alignments, each of which have different phylogenetic trees. So to give you an example, one data set we've been working with recently is from Wong et al. 2014. It's peripheral blood samples from 27 subjects, all of them healthy, and between 3,000 and 9,000 unique sequences per subject. So if you take those sequences and cluster them to try to separate out clonal lineages, you'll find quite a few lineages. And one large issue is that 88 to 96% of these clonal lineages will have only a single unique sequence in them, which isn't exactly a very interesting phylogenetic tree. And less than 2% of these lineages, depending on the subject, have at least 5 sequences. So really within these data sets, you're dealing with a lot of very small trees. So you have this paradox of the fact that the information contained within the repertoire is vast. There's a huge amount of sequence information within a particular B-cell repertoire. But within each individual lineage, there's often very little information. So our approach for dealing with this fact is to use hierarchical phylogenetic substitution models. Whereas in standard approaches, we would take the single multiple sequence alignments from each clone and estimate a set of parameters for that clone and a separate phylogenetic tree for that clone. We instead estimate one large substitution model in which model parameters are shared among all of the lineages within the repertoire. This gives us a lot more power to accurately estimate the substitution model parameters. So we effectively read in all of our sequence data from a particular repertoire at once and estimate all of the substitution model parameters and phylogenetic tree topologies for an individual's repertoire simultaneously. And this has a number of advantages, particularly when it comes to parameter estimation. So here we have a fairly simple simulation analysis where we have taken phylogenetic tree topologies from a particular individual's repertoire and simulated clonal sequence data sets using a neutral model of somatic hypermutation targeting, and then tried to re-estimate these substitution parameters from our model. And what you can see is that when we use individual models on very small clonal lineages of less than 10 sequences, the parameter estimates are essentially meaningless. They have extremely high percent errors. Once we get more sequences, the percent error goes down, but it's still not great. If we use a hierarchical model using the information in all of the lineages, we're able to get these percent errors down to single digits in most of the parameters we estimate, which is a massive improvement. And these are on fairly small data sets compared to modern data sets that are being generated. So this should give us even more power in the future. And this gives the intriguing possibility that we could actually use phylogenetic substitution model parameters to characterize the repertoires of individuals. So here we have parameter estimates for 27 healthy subjects. The top panel shows parameter estimates for particular hotspot and cold spot motifs in somatic hypermutation. And as you can see, they're generally conserved among these 27 subjects, or at least the hierarchy of GYW, WRC hotspots. We also have the DNDS measures for different regions of the antibody sequence. So framework regions are used for structural purposes and are generally under more constraint, whereas CDRs are antigen binding and are usually more neutral. And we can see that there's actually quite a lot of variation among subjects in the selected constraint on the CDRs, which leads us to ask, does this vary by disease state, age, or sex? And unfortunately, you're just going to have to wait to hear the answer to that for another time. But if you would like to answer those questions for yourself, all of this software is currently available on Bitbucket through IGFIML. You can get it through that URL. If you want to talk to me about any of this, I have a poster, B843, this evening. And big thanks to the Pharma Foundation for funding my research, as well as my supervisors at Yale and Oxford. Thank you.
Thanks.